All right, today we're diving deep into one of the most remarkable organisms in all of parasitology, the schistosome, also known as the blood fluke. We're going to get into its really unique biology, the devastating disease it causes, and the incredibly intricate relationship it has with its hosts.
And we're starting with this absolutely incredible image. What you're looking at is a scanning electron micrograph of a male and female schistosome. They are literally locked in a permanent embrace. The larger male holds the more slender female inside a special groove called the gynecophoric canal for their entire adult lives. This pairing, this lifelong commitment, is absolutely key to the parasite's success.
But this fascinating biology, of course, has a very dark side. Schistosomiasis, the disease these worms cause, infects an estimated 240 million people across the globe, mostly in tropical and subtropical areas. It's a huge public health problem that traps entire communities in a vicious cycle of poverty and chronic illness. Okay, so here's our roadmap for today. We'll start by properly meeting the organism itself. Then we'll trace its really complex life cycle. After that, we'll look at the pathology, how it actually makes people sick. Then we'll cover diagnosis, and we'll finish up by looking at treatment and some of the serious threats on the horizon.
Right, let's get into section 1 and get a formal introduction to our parasite, the schistosome.
So, taxonomically speaking, where do schistomas fit? Well, they're in the phylum Platyhelminthes, so they're flatworms, and more specifically, they're in the class Trematoda, which are the flukes. The genus name, schistosoma, is just brilliant. It comes from the Greek schisto, meaning split, and soma, meaning body. And as we just saw, that perfectly describes that unique groove in the male worm, which is the defining characteristic of this entire genus.
Now, to really understand this parasite and the disease it causes, you have to follow its journey. The schistosome has what we call a complex digenetic life cycle. That just means it relies on two completely different hosts to survive. And this is fundamental to how it's transmitted and the disease it causes. So this is the whole shebang, a diagram from the CDC showing the entire life cycle. It might look a little complicated at first glance, but it's actually a beautifully tuned process that alternates between a definitive host, that's us, humans, and an intermediate snail host. So let's break it down, starting with the eggs, which you can see at stage 1, being shed by the human. It all starts with environmental contamination. Eggs are passed from an infected person, either in their feces or their urine. Now, if those eggs make it into fresh water, they hatch, and they release this tiny, free-swimming larva called a marasidium. The only job of this marasidium is to find and burrow into a very specific species of freshwater snail. And this is critical. Without that specific snail, the entire life cycle is broken.
This slide shows a really critical transformation. On the left, you've got a mansoni egg, that's the diagnostic stage, and you can actually see the developed marasidium coiled up inside. But after its journey through the snail, it transforms into what you see on the right, the cercaria. Just look at that distinctive forked tail. This is the free-swimming stage that is actually infectious to us.
Now, what happens inside that snail is just incredible. That single marasidium undergoes a massive process of asexual reproduction. It develops into these structures called sporocysts, which basically turn the snail into a parasite factory. A single infected snail can pump out thousands upon thousands of those fork-tailed cercariae back into the water. And then, when a person comes into contact with that contaminated water, the cercariae actively burrow right through their unbroken skin. And that's it. A new infection is established. Okay, so once they're inside the human host, the parasites mature, they find a partner, and they migrate to the veins around the gut or the bladder. But here's the crucial thing, and if you remember one thing about the pathology, make it this. It's not the adult worms that cause the severe disease. It's their eggs. The real damage is done by the eggs. A female worm can produce hundreds of eggs every single day. Some of them are shed, but a huge number get swept away by the bloodstream and become trapped in host tissues. This triggers a massive inflammatory immune response called a granuloma. For species like S. mansoni and S. japonicum, this happens in the liver and gut, which leads to scarring or fibrosis, and a life-threatening buildup of blood pressure in the portal vein. For S. haematobium, the eggs get stuck in the bladder and urogenital tract. This causes chronic inflammation, scarring, and, critically, it's classified as a group 1 carcinogen because of its strong link to bladder cancer.
And this slide just illustrates that connection perfectly. On the left, you see the macroscopic clinical result of chronic intestinal schistosomiasis, hepatosplenomegaly. That's a massive enlargement of the liver and spleen caused by that high portal blood pressure. And on the right, you see the microscopic cause, the eggs themselves, trapped in the tissue, completely surrounded by the host's own inflammatory cells. It's the host's reaction to the eggs that drives the disease.
So, given this really complex disease process, how do you actually go about diagnosing an infection, either in a clinic or out in the field? Well, that brings us to the diagnostic toolkit.
The gold standard, believe it or not, is still direct detection of the eggs under a microscope. We use a technique called CATO-CATs for stool samples or a simple filtration for urine. These are great because if you see an egg, you have a definitive diagnosis of an active infection. But their sensitivity can be low in light infections. Then you have immunodiagnostics, like antigen tests, which are good for detecting active infections, and antibody tests, which can stick around even after an infection is cleared. And finally, you have molecular methods like qPCR, which are super sensitive for detecting parasite DNA, though they still usually rely on having some eggs in the sample.
Okay, this brings us to our final section, how we actually fight back against this parasite and the really serious challenges that we're starting to face. For decades now, the entire global control strategy has relied almost exclusively on a single drug, praziquantel. It's safe, it's effective, and it's cheap, which makes it absolutely perfect for these huge mass drug administration programs that treat millions of people every single year. And this slide shows you exactly what the drug does. It's amazing. On the left, you have a healthy worm just chilling. On the right, that's a worm after it's been exposed to praziquantel. The drug causes a massive influx of calcium ions, which leads to spastic paralysis. The muscles just lock up. But crucially, it also causes severe damage and blistering to the worm's outer surface, its tegument. This exposes parasite antigens that were previously hidden, which finally allows the host's immune system to see the worm and attack it. It's a fantastic tool, there's no question. But the one question that looms over the entire field, the thing that keeps researchers up at night, is this. With so much riding on a single drug, how long can we really count on it? And there are several really worrying signs. First, you have this immense selective pressure from treating millions of people over and over. That's coupled with constant reinfection from untreated water sources and people, which creates the perfect conditions for resistance to develop. On top of that, we're already seeing reports from the field of the drug not working as well as it used to, and we know that the larval stages of the parasite are naturally less sensitive to it. All of these factors are creating a perfect storm for the selection and spine of praziquantel-resistant worms.
So this leads us to a really critical conclusion. The fight against this ancient parasite is entering a brand new phase. The next chapter isn't just going to be about mass drug administration. It's going to be defined by our ability to understand the molecular basis of drug resistance and to develop the next generation of tools we need to stay one step ahead of this remarkable parasite.